Okay, so great to see everybody here. Thanks for joining us today. So just to give a little bit of background, my own background, the highest level of science, or at least of biology, that I took was grade 9 in high school. And I remember when we got our diagnosis, I guess about 6 years ago now, I would go to these scientific meetings and conferences and try to understand what researchers were saying. And like just so much of it was going over my head. And then there was one conference that we hosted here in Toronto. And we had some researchers there. And one of the researchers, Dr. Amy Ramsey, who's going to be speaking in another one of the sessions, I think in the third session, she talked about research that she had done where she was able to take a mouse with GRIN and basically build a sort of off switch in the mouse so that the mouse would grow up with a GRIN1 variant and then turn off that variant, so the mouse's brain would sort of go back to a more typical brain when the mouse was adolescent or adult. And what she found is that when she did this, the mouse's brain, the wiring in the brain would sort of start to return to more like a typical mouse, but also the mouse's behaviors would improve. So the mouse would become more social, more intelligent. Like a lot of the things that we saw changed with GRIN would become much closer to real mice. And so that really got me excited about, that was one thing I could finally understand and got me more excited about GRIN and doing research into GRIN and eventually into all of GRIs. But I started by taking all of the sessions from that Toronto conference, because we had recorded them, and like watching them. And every time there was a term I couldn't understand, I would like pause it and go look up on Wikipedia. And so often like the Wikipedia entry I couldn't understand. So then I'd go look up other things. And so it would take me like, 4 or 5 hours to get through a 30-minute talk. And I still didn't fully understand it, but gradually I learned more and more. And, this is the one thing that we hear so often when we put on these conferences, is that families say that they really appreciate everything they learned. They just wish they could understand more of it. And so we were originally, when we were hoping to do this year's conference in person in Boston, we were going to have the first afternoon set aside for this GRIN university session. And when we realized we had to do it online, we decided to do it this way. So we have 3 sessions this week, and then every 2 weeks leading up to 2 weeks before the conference. So one thing that I want to do just before I turn today over to Megan is, does everybody know how to use the chat function? So if you could go to the chat and don't press enter yet, but go to the chat and just type for yourself what the sort of highest level of biology that you studied was. Did you do, which grade was it in high school, or did you do anything in what grade in high school, or did you do anything in university or post-secondary, university or college or post-secondary education? So type it in, but don't press enter. We'll all press enter at the same time. Just type it in, don't press enter. Does anybody need more time? Has everybody entered? Has everybody put it in?
Yes, I'm still typing.
Okay. Okay. So press enter now. Okay, so it's a mix here: grade 9, university, A levels, high school biology, college biology, now in medical school, learning more and more each day, A level, Scottish higher, high school age 16, anatomy, physiology in college. So yeah, a real mix. So it's great, and we've really asked the researchers to try and keep things simple. But if there's something that you don't understand, you can interrupt them, or you can let Megan and I know, and we'll interrupt them. We really want to make sure that because this all builds on each other, we really want to make sure that everybody can follow all the way through. So thanks for joining us. Welcome, and over to you, Megan.
Hello, everyone. I hope you can hear me now. So I just wanted to give an overview of this session. So essentially, as Keith mentioned, this is a three-part series. So we are starting with genetics and GRI disorder today. Then we are going to be proceeding to neuroscience, GRI receptors, and personalized research on March 26th. And then on April 9th, we're going to close with GRI treatments and cures, which is just a couple of weeks before the conference. So hope to get everybody engaged and give everybody a good baseline to build off of as we move towards the conference and beyond. Let's see. Okay, so today we are starting with genetics and GRI disorders. So this session, we hope to provide the basics of genetics, how it relates to GRI disorders, and we're hoping that you'll be able to take information away regarding prevalence, symptoms, treatments, life expectancies of GRI disorders, and what life looks like for GRI adults. And so we're going to go until about 12:30. After each speaker, we're going to have a few minutes for questions. So we're going to start with Eriskay Liston, who is a genetic counselor at the Hospital for Sick Children in Toronto. And then Dr. Johannes Lemke is going to talk about the genetics of GRI disorder, and he is a medical geneticist at the University of Leipzig in Germany. So without further ado, Eriskay, if you want to share your screen.
Sure. Okay. Can everybody hear me okay? Okay. I had a mild panic because I had to switch up computers at the last minute. I had an issue with my audio, so I'm glad everyone can hear me. So just give me a second here. All right. Can everybody see my screen? Let's try to get it into, there we go. That's better. Perfect. Okay, great. Well, it's a pleasure to be here. Thanks so much for inviting me to speak with the group, Keith and Megan. It's really my pleasure. I'm really happy to take questions as we go along. I know, Megan, you had some time allotted at the end for questions, but it's smaller group, and sometimes it's if there's a concept that's coming up that you want to just kind of interrupt me and stop me in the moment, that's fine. Sometimes that's an easier way to do it. So up to you guys, but I can't see everybody, so just feel free to interrupt. And today's agenda is really gonna, as Megan already said, we're kind of gonna do a little bit of a background on genetics. So I'm calling it Genetics 101. Just sort of scratching the surface and hoping to really help you all, be able to understand the information that's coming, over the next few courses that are being held as part of this GRI University. We're going to talk a little bit about, inherited and non-inherited types of diseases and the sort of more classic forms of inheritance. We'll talk about the, genetic report that some of you may have, seen or looked at or read, and, just try to give a few, orient you a bit to those reports and help you understand them better. And then have some time at the end to ask to answer some of your questions, I hope. So going back to the basics, and again, I know there's sort of a degree of level of backgrounds in biology. So we're just gonna, start at the very beginning. This is what we call a picture of a karyotype. And what a karyotype is basically is it's a picture of our chromosomes. And really, I look at this and I think to myself, this is really the instruction manual for our bodies. It tells our bodies how we grow, how we develop. It contains all that good information. So the sort of clump of chromosomes on the left is really what you would see if you kind of opened up the nucleus of a cell. And then on the right, what you see is they're all organized into pairs. And what's important to note here is the numbers. So you can see that there's 22 pairs. And then at the bottom right there, you see there's, you know, 2 X's, and then there's nothing on the Y. So this indicates here, this karyotype that we're seeing is a female karyotype, because females have 2 X chromosomes. If it was a male karyotype, you would see 1 chromosome in the X and 1 on the Y. And so this is kind of, again, what the first step of kind of orienting ourselves to what is genetics. Now, I'm going to break this down a little bit. So we have, you know, trillions and lots of cells in our body. And just imagine this might be a cell, you know, in the brain. And, you know, if you opened up, like I mentioned, that, you know, cell in the brain, you would see that and most cells in our bodies have a nucleus. And if you opened that nucleus up, that's the part in the middle, you would see these chromosomes. And the chromosomes, I kind of like to think about as sort of the chapters of our instruction manual. If we take it back to that analogy, you know, if you're given a manual for your car, for example, it's going to be broken down into sections or chapters. It's the same with our genetics. We have this instruction manual. Our chromosomes are kind of like the chapters. And then if you sort of look very closely at a chromosome and kind of pulled it apart a little bit, you would see that it's, you know, filled with lots and lots and lots and lots of genes, you know, thousands of genes on each chromosome. And the genes are sort of like the sentences or the paragraphs within those chapters. And so it's a segment of DNA, and DNA is just, is really like our code, okay? And that's made up of, amazingly so, actually only a combination of 4 of these letters, or what we call nucleotides, which are adenine, thymine, guanine, and cytosine. And it's the different combinations of those letters or those nucleotides that actually make each gene, you know, unique, and tells the gene, or tells, you know, our bodies about, you know, a protein that we need to make. So each arrangement, specific arrangement of those letters goes into making, you know, these, what we call amino acids that link together to ultimately make a protein. And the proteins are really, again, the sort of things in our bodies that tell our bodies how to develop and grow, and so on. So that's kind of the way that this works. And amazingly, we have, you know, 6.4 billion base pairs. So these base pairs are the connection of 2 of those nucleotides that come together. And so when you look into a cell, like in the brain, for example, where there's, you know, 2 copies of every chromosome, like I showed you in the picture before, if you break that down over, that's 6.4 billion base pairs. So lots and lots and lots of base pairs. And so just to kind of help you sort of understand these nucleotides and letters that we're referring to, if you think about, you know, our language and our, you know, the human or English language, or any language, for example, really what makes sense of that, or how we make that, is through letters, right? So the alphabet there that you see, and different arrangements of the letters make different words. Same with music. When you think about how do we get, and you know, to play a beautiful piece of music at the symphony, for example, that's because of the arrangement of the notes. So the notes, the way in which those notes come together, you know, dictate how music might sound. So same with DNA. When we think about these 4 letters here, or nucleotides, the way in which they're arranged will tell our bodies how to function and how we grow and develop. Any questions as you know, because I think this is sort of a foundational piece that's important. I don't see any hands, so I'm just going to keep going. And the way that these nucleotides come together is, as I mentioned before, they come together in threes. Okay, so they join together as a group of 3. And depending on how they've joined together, they will make a unique or different amino acid. And these amino acids are really what we call the building blocks for our bodies, and they come together, and the specific sequence of amino acids will make a protein. So if you change, you know, one of the amino acids in that sequence, it could completely change the protein, okay, and will change the way that it's made, or, you know, the type of protein that's made, or how functional that protein is. It's kind of like when you build a tower, you know, if you switch the bricks for blocks of wood instead, you know, that may work, but it might not be as solid, for example. Whereas if you, completely remove the bricks and took the bricks out of that foundational structure, that tower is not going to be able to be made. It will crumble. So with GRIN disorders, this is just to show you, in this chart, you can see examples of different names of genes and types of genes and the different proteins that gene encodes for or makes. So that's really the more important piece to see, is that, again, that concept of the sequence or the code that we get from the gene, depending on the letters or the nucleotides that come together, a different protein is going to be made. So for GRIN disorders, or for GRIN, we know that it's really important that there is this, that 4 of these proteins come together to make this specific type of receptor. And this receptor, in what it does in the brain cells, is it really, it helps, it's like a channel. It sort of opens up the membrane in a brain cell and allows things to come in and out of that cell, and allows for communication between the cells. And so if this receptor, if the proteins don't come together properly, it can alter the function and the ability of that receptor to do its job, such that things can't come and go out of the cell in the way that it normally should, for example. And so in the case with GRIN disorders, we know that if the receptor is altered in a certain way, communication between the brain cells doesn't happen as it should, and that can therefore, be responsible for the symptoms that we see associated with GRIN disorders. So just a quick pause to review, just the key points that I went over is that there's many letters in the human code, so 6.4 billion nucleotides. And that really comes down to arrangement of 4 different nucleotides, the A, C, G, and T. And that every word is about 3 of these letters long. And those are what we call the amino acids. And then each of these amino acids tells our body, or these letters come together to make these words to tell our bodies how to work and how to function. And they come together and are sort of like the building blocks. And I think that's the important piece, is that they're really that foundational piece to build certain things like the receptor that we talked about. And there are 4 proteins in every NMDA receptor, 2 usually of the GRIN1 and 2 of the GRIN2 proteins, which are coded by the different GRIN genes. So I want to shift gears a little bit to talk about how genes and variants and within genes are inherited. So if we, I'm going to focus really on the sort of core, sort of classic types of inheritance that you might have heard of before, and just highlight here a few different types of conditions or diseases that have these types of inheritance patterns. So the first one that I'm going to talk about is called autosomal dominant inheritance. And when I say autosomal, if you think back to the picture I showed you at the very beginning, those 22 pairs of chromosomes, those are the autosomes. Okay, they're the ones that aren't specific to the gender of the individual. Okay, the sex chromosomes are the X and the Y that I pointed out at the beginning. So this is an autosomal dominant classic kind of inheritance pattern. And what you see here is that you see a father and a mother, and then the potential options of children that they can have in terms of, what type of genetic trait is inherited. So if you look at on the father, he's got his 2 chromosomes, because remember, we have 2 copies of every chromosome. Same with the mother. And the little squares on the chromosomes are highlighting that there is a trait or a gene that has a variant or a difference in it. And that's the one that we see that's white. And the darker one, or the blue one, is the unchanged or, sort of normal variant. So the mother has 2 copies on both of her chromosomes. She's got 1 on each chromosome that are unaltered and kind of what we would call normal. And the father on 1 of his chromosomes has a change in his gene. And some people refer to this also as an allele. So you may hear that term as well, as an allele or a trait. But what I'm referring to is the specific variant. So when the mother and father, are having children, they only pass on one copy of their chromosome, because remember, the child can only have two. So one is going to come from the mother and one is going to come from the father. So when they have children, it's really a 50-50 chance of which chromosome or gene or variant they're going to pass on. Okay, so you can see from the options below, the father is either going to pass on to his children the chromosome that contains the unchanged or normal variant, or he's going to pass on the chromosome with the variant that's housed within it, in that gene. Okay, is that a question I'm hearing, or is it just, I hear it, okay. I just heard something and I just don't want people to feel inhibited. Please speak up if you have a question. So 50-50 chance. And so you can see below that, you know, of the children that they may have, half of them are in white, meaning that they've inherited that change, and half of them are not, right? Because the other two would have inherited the chromosome without the change. And of course, the mother only has a chromosome with that has, you know, the normal copy of the gene. So is always going to pass on that normal copy. So that's a classic sort of inheritance, autosomal dominant inheritance pattern. And some of the conditions that you may or may not have heard of that exhibit this type of a pattern of inheritance are Huntington's disease, something called neurofibromatosis, Marfan syndrome, and familial hypercholesterolemia. So not really important to remember all the names. It's just sometimes people may be familiar with that type of condition, and therefore the type of inheritance seen there. So this here is something called autosomal recessive inheritance. And so what's different about this pattern is that you can see that in this case, both the mother and the father have one of their chromosomes that has a genetic change in the copy of their gene, which is again highlighted by the white little square on the chromosome. So here, this is a scenario where you know, the parents generally are what we would consider unaffected. So generally, they don't have the condition, but when they both happen to pass on the chromosome with the genetic change to their child, that child then becomes affected by the condition, okay? So in this scenario here, you see that 25% chance for their child or their future children to have this condition, because it's again a 50-50 chance for each of them to pass on the chromosome that has that genetic change in it. We do know that there are, you know, it's uncommon, but there are some types, there are some families that we know of that have GRIN disorders that have this type of inheritance pattern where it's been found that the parents have been carriers, but this is uncommon. Most of the time, and I'll get to that in a minute, with GRIN disorders, it's a different type of inheritance where it's actually generally not inherited. It's something called de novo, and we'll talk about that in a minute. But in this case, the variant that's seen in the gene that's you know, can be causing the disease, it can be either the exact same variant that the mother and the father have in the gene, which is called a homozygous type of situation. So that affected daughter on the bottom right may have inherited the exact same variant from both the mother and father. And there are certain conditions where it's quite common that we see, you know, in certain families that the mother and the father are carrying the same variant and pass it on, so that the child has what's called a homozygous variant, meaning the exact same variant on each copy of their chromosome. Often that's not the case. Often the case is where the mother and father happen to have a variant, a different variant, but in the same gene. So that's a compound, that's called a compound heterozygous scenario, where the mother and the father just happen to have a change that affects the gene, but it's actually a different type of change in both the mother and the father. And then in this scenario, you also see that 75% of the time, this couple or this mother and father would have, you know, a child that is unaffected, but 50% of the time, their children will be carriers as well. So hopefully that makes sense to everybody. Some of the more common or known disorders that have autosomal recessive inheritance patterns are cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and again, as I mentioned, some form of GRIN disorders. The sort of last type that I'm going to talk about, the sort of classic inheritance, is X-linked recessive. And this is just a little bit different from the others because this is when the change is found on the X chromosome. And so as you can see from the picture on the left, where you have a carrier, a mother who's got a change on her, one of her X chromosomes. And remember, females have 2 X's, males have 1 X and 1 Y. There would be a 50-50 chance, again, similar to sort of in the, dominant scenario of which, gene, which chromosome she's going to pass on. So if she has a male, a boy, and passes on the changed X chromosome, that boy would be affected because he only has 1 X chromosome. He doesn't have another X chromosome to sort of mitigate the effect of that changed gene. So he would have be affected with the condition. Whereas if the woman passes on her changed gene to a female, so the other X chromosome comes from the father that is unaffected, she would be considered a carrier, but not considered affected with the disorder. In contrast to that, when a man who is affected with an X-linked recessive disorder, he would have unlikely to have, an affected daughter, or really doesn't have an affected daughter because there's always going to be that other X chromosome coming from the unaffected mother. So all of his daughters would be carriers, but unaffected. And because when they have males, he can only pass on the Y chromosome, they're unaffected and can't be carriers. So that's sort of the difference between when it's a female who's carrying the condition versus an affected male who has an X-linked recessive disorder. And different types of conditions that have X-linked inheritance patterns are one that you might have heard of is Duchenne's muscular dystrophy, Fragile X syndrome, or hemophilia. So GRIN, we know mostly are the result of what's called a de novo or new variant or mutation that is not generally inherited from parents. So that's the most common way that when we do testing for GRIN disorders that we find that it's really not inherited from parents. I will say there is a caveat to that in that sometimes, a parent can have a change in one of their germ cells, so not in all of the cells of their body such that they are affected with the condition, but can have a change in one of their germ cells such that there is, they would be considered to be sort of at risk of having, additional children with this condition because they can pass it on through their germ cells. And this is something called germline mosaicism. It's quite rare, but it is something that can happen. And so generally when we find that there's a de novo change or a new mutation in a child with a GRIN disorder, we do offer testing for future pregnancies or children in the family because of the chance of that happening. But this is generally the case with GRIN. There's a number of other conditions where we generally see a high number of these de novo events happening. Noonan syndrome is one, Rett syndrome, and Dravet syndrome, which are other types of neurological conditions where generally we see, in children with genetic variants that are causing this condition that the parents do not have it. And so just to sort of, highlight that, the difference between inherited conditions, classically inherited conditions and others, there are other types of genetic inheritances that don't follow these sort of classic patterns. One I want to talk about is that sort of the multifactorial inheritance where a condition may be considered, genetic or have you know, changes in genes that are associated with the condition, but that it's maybe not just due to one gene or one variant. There may be multiple gene changes or multiple variants that contribute to the risk for this condition to happen, as well as potentially environmental influences. And we call that multifactorial inheritance. And a classic example there would be something like autism or schizophrenia or cerebral palsy, which, you know, we're learning more and more about these conditions as we do more, you know, genetic discovery and testing. So we do understand that there's a fair amount of genetics and genetic variation that's related to these conditions, but don't follow those same patterns of inheritance that I spoke to you about before. And then non-kind of genetic conditions like, you know, infectious. I use COVID-19. I think we're all familiar with sadly, with COVID-19 now. But malaria, chickenpox, these are different types of conditions that are, you know, not related to, you know, inheritance or passing on of genes, and teratogenic. So things that are, you know, conditions that happen because of exposures in the environment during the development of pregnancy. So various different types of conditions. We know that GRIN is a genetic disorder, and we're still learning about, you know, different types of genes and variants and how that impacts, you know, and has impacts the development of children with GRIN. So a little bit now I want to switch gears again to talk about the reports that some of you may have seen associated with, you know, having testing for GRIN disorders. I'm going to focus a little bit on exome sequencing. I think exome sequencing is probably one of the more common tests that people might have had, you know, to lead them to a diagnosis of a GRIN disorder. More frequently now, depending on where, you know, the testing is done and in what center, you know, we're starting to see something called whole genome sequencing, which is, you know, even more comprehensive and looking at more of our genetic makeup. But I think probably exome sequencing is the one that people are more familiar with. And basically, this is the process of really reading and understanding that genetic code that we talked about at the very beginning. You know, taking all of the chromosomal material, looking, you know, pulling it sort of apart and looking at all of those letters and the sequence of the letters and looking for changes in those letters, or that are changes from the sort of usual code. And it's quite a complicated process. When we think about exome sequencing, you know, it's actually only looking at a small portion of all of those, you know, all of that genetic code. Because really, when you break down, I don't want to get too detailed, but when you break down the gene, there's parts of that gene that we know code for proteins that are really important in coding proteins, and other parts that don't code for anything as far as we know at this time. And so this technology kind of takes out those parts that don't code for the proteins and focuses just on the protein coding section of that gene. And when you take out all that other stuff, it actually ends up only being about 1% of the genome. So it's not looking at everything. In contrast to that, whole genome sequencing, which is a newer technology that's becoming more and more available in clinical settings, actually looks at that whole gene, even the parts that we know don't really code for protein, because we're learning more about that part, and we're learning that actually changes in those sections can impact the parts of the gene that do code for proteins. So using whole genome sequencing allows us to increase our diagnostic yield in a way that we weren't able to do before. So depending on the setting, usually when you apply, you know, when you do exome sequencing for a family, you might have a diagnostic rate of about 25 to 40%. The other thing with exome sequencing, I'll touch on briefly, we don't have the opportunity to really get into it, is, you know, that you can get additional information from exome sequencing besides the information that you were looking for around a specific diagnosis. So sometimes you can find out that there might be changes or variants in other genes that might be predisposing, you know, a child or family member to other types of risks. So there's things to consider with this type of technology, and highly valuable to undergo this type of testing with genetics professionals who kind of understand what types of results you can get out of this and help families understand the types of results that they want given back to them. So this is an example.
I wanted to ask a quick question that we got from the chat. So someone was asking about next generation sequencing and if that is a type of exome sequencing.
Yes, it is. A great question. You know, it's just different terms that people use. So next generation sequencing is sort of the technology that's used, and exome sequencing is an example of next generation sequencing. Some people, the way it's sort of evolved with next generation sequencing is they used that technology, but they started using it in clinical labs kind of in what we would say like panels of genes. So very sort of targeted use of that technology. They would say, okay, we only want to look at gene, 10 genes or 20 genes or even 100 genes that they sort of would choose and then apply that technology to. But now we can, next generation sequencing often now is something that we use really, and whole exome sequencing is sort of a really good example of next generation sequencing.
Thank you.
Does that answer the question?
Definitely. Good to go.
Okay. So if I just want to focus on a couple of the types of mutations that we can get from, or that we can see from this type of technology. So, this is sort of what the machine would see on a basic level is would sort of, open up the DNA and look at the code. And you can see that, in the code, there's like how I mentioned, there's groups of these 3 letters together and they code for this protein below. And if you just change one of those letters in that sequence, it could completely change, the sequence of amino acids and then ultimately produce, a protein that's either, a less functional or non-functional at all. This is an example here of a missense mutation where it's just one specific change on, in the code. So just to kind of put this back into the context that we were talking about with the, instruction manual, and we go back to this, idea of chromosomes being like the chapters in a book and genes being kind of like the sentences. If you look at like a word from one of those sentences, this would be an example of a missense mutation where you're looking at, the word red, and instead of red, the E is substituted for a D, and you end up with a word that you don't really understand or doesn't make sense. You can also end up with a sentence that doesn't make sense. So the car was red is the original, intended sentence. And then if there's a deletion or a change of a specific word, it completely changes the meaning of that sentence. The was red, it doesn't really mean anything. So below, you can see kind of the different examples of types of mutations or variants and the effect that they might have on the word or sentence, or hence the genetic code and then the production of a protein. So again, with the missense mutation, instead of the car was red, you might have something that says the car was hat or the car was red, which doesn't really make sense. The thing I want to point out is that dependent on the type of mutation, even with the missense mutation, sometimes it can be still sort of understood. So sometimes a protein can still kind of be made, but just might not be as functional. Whereas in other types, like for example, in the missense mutation box there, you see the car with hat, that it doesn't make sense at all, and we can't really understand it. The car was rud, we might be able to sort of interpret that this sort of means the car was red and that there was a mistake. And this is just to say that different types of missense mutations might have different impacts on the gene and the function. So we can't make sort of statements that like all missense mutations are going to result in the same kind of outcome. It really depends on the variant and the impact it has on the ability of that protein to be made. I focus a lot on missense mutations because this is really probably the most common type of mutation that we see in GRIN disorders. Insertion mutations, I won't spend too much time here, but just so you can see the different types of mutations and the types of impacts they have on the development of the protein. So I wanted to just show you, and many of you who may have had results come back might have a different type of, looking report, depending on the lab that was used. They might organize the information in different ways. But what I think that is important to sort of see here is basically the categories of information that you can get. So this is just one example of a result and the type of language that the reports use. So, and you see here in table 1, pathogenic variants in disease genes related to the clinical phenotype. So you might look at that and what is a pathogenic variant? What does that mean related to clinical phenotype? So the way that labs are often categorizing variants is in different levels of sort of the effect that they've interpreted to have on the protein. So we know when we call something pathogenic, we know that that's altered the function of the protein enough so that we're saying that that variant is actually causing the condition. So that's what we mean by pathogenic. I'm gonna, in another slide, I'll go over sort of the different levels of what the variants can be. But in this case, it's really important to know when you get the report back, what they're calling that variant. Are they calling it pathogenic? Are they calling it a variant of uncertain significance? And so on. So that helps you kind of understand the meaning of that variant and the significance of that variant. Usually in the report, they'll sort of link the variant and the gene to a specific condition. And that condition may be referred to in different ways. Sometimes a specific name of a condition will have multiple different ways of being explained as to what it is. So in this condition, for example, it was a GRIN1 result, but the disease was referred to as mental retardation autosomal dominant 8. It was a term that was coined by before there was an actual understanding of GRIN1. So sometimes people will see these names in a report and be confused as to why they're referring to it in that way. And that's why it's really important to go through these reports with a clinician, a geneticist or a neurologist or somebody who's really familiar with the information and can really explain it to you. It's important to see the inheritance pattern. And I wanted to just point out that, they're calling this autosomal dominant, but then if you look to the end of the report or under the comments, they're explaining that this is a novel variant. So neither the mother nor the father has the genetic change, so they're calling it de novo. The reason it's being given the autosomal dominant inheritance pattern is because in this individual now, this is a genetic change that is in all of the cells of their body. So it's considered autosomal dominant. That would be passed on in the way of a autosomal dominant inheritance pattern. So sometimes that can be confusing for patients as well. I think the other important piece is just for some parents is to kind of understand what the change was. So looking in the section of the report, you can see they call out the name of the gene, where the position is on the chromosome, the exact location of, what exon it was in, and the nucleotide change. So here you can see it was at position 1858, and it was a change of a G to an A. And that was the actual change that caused, that was deemed to be pathogenic variant to cause the condition. So I think I kind of went over this mostly. It's just to highlight again the important pieces that parents like to know about in the report, and to make sure that there's a clinician or somebody that understands the information and can really translate it and explain it to the families. And then this is just a couple of different examples of other types of variants that we've seen both in, in many of the different types of GRIN genes. So missense mutations, insertions. A frame shift is really just when it, shifts the the organization of the amino acids and then into a different position. So, and sometimes that can cause the protein to actually not be made at all. So that's why frame shift stops. So it actually stops the production of that protein. So it's not made, it's not that it's made and, you know, inefficient, it's just not made at all. And then there are some deletions and insertions as well. And so just very briefly, you know, the labs that do this kind of testing use different levels of evidence and types of evidence to assign the variant, put it into a classification of either pathogenic, as we saw in the previous report, likely pathogenic, so that there's, you know, enough evidence, or maybe it's being reported before, but not enough to really call it pathogenic. A variant of uncertain significance, I think, is an important one I just wanted to highlight, because as we do more and more testing and genetics testing and genomic sequencing, we see more and more variants of uncertain significance. And over time, these sort of variants that we don't know exactly how they're impacting the function of the protein and therefore impacting, you know, our bodies, you know, we're going to probably learn more about them, and the significance of a variant can change over time. So sometimes you can find a variant of uncertain significance that your doctor might, you know very well, you know, the gene and how it sort of works, and might say, I think we need to kind of watch this over time. And it may go from a level of uncertain significance to pathogenic. Even sometimes in the course of a year, that interpretation can change. And so for people who end up with, excuse me, changes on a genetic report that are uncertain, it's really important to check back in with the specialist to understand how the evidence that's being collected about this variant could potentially change the interpretation of this report. Sometimes it can go the other way, where there's a variant of uncertain significance that, you know, is thought over time as we learn more about it to be less significant, and goes into the direction of being something that's likely benign, or benign meaning not related to the condition that we're looking to diagnose. So I just wanted to highlight that, because I think, again, it really emphasizes the importance of the relationship between the clinical team, the lab, and the patient and family, and having sort of these ongoing discussions for, you know, around the results of a genetic test. And can be very confusing and challenging for parents and families to understand. So I think, hopefully, I think I've gone over time here, so I'm very sorry, but if we don't have time for questions, I'm happy to stay on to the end and answer them then.
I do think we had one question in the chat. So someone was asking about how do you know the type of gene mutation? And they noted that their report didn't mention the type of mutation.
So it should mention the type of mutation in the report. I would say that's a question, you know, almost all genetic reports will have that information in there. It's just sometimes buried. It's sometimes not as you know, as obvious as the one I showed you, where it's in a little chart where it kind of breaks everything down. So I think the important piece there is to, you know, take that back to your clinician and, you know, ask them to walk you through it, because it really should be there. That's how they arrive at the molecular diagnosis, is by, you know, sort of finding that change and then classifying it.
Tina may be asking about it not saying missense or that kind of thing. And I think that wouldn't necessarily be spelled out in a genetics report. You'd have to decode it from the letter sequence. So
but that's exactly it. My letter doesn't say anything about that. Whether it's missense or...
Yeah, I don't know if you want to pull up that slide where you have the different variants just for a second, Eriskay, and just show...
So that's also something that, I mean, again, I think if it's not actually on the report, it should be something that the clinician should be able to walk you through. But let me just pull up the, was it like this one, for example? Yeah. Like, so, and again, the way that, it's funny because there's some level of standardization across clinical labs and sort of a nomenclature, we call it, or like way in which they write these that should be consistent. But sometimes you'll see, you know, they'll use the sort of code for the, you know, the amino acid that was changed and the position. So this is that position, you know, 117, a proline was changed to a leucine. And so we know that that's a, you know, a type of missense mutation, but depending on the way that it's being written in the way the, you know, the nomenclature that the lab uses, sometimes it's easier to kind of figure that out. Like if you look at the one here, for example, it's written in a different way. You know, they sort of showed the position of the nucleotide, you know, at position 1858, we changed a guanine to an adenine, which is, you can also translate to, you know, something like this, but it's not in the report that way. So if you actually have that information now, we can look at it, or I can look at it right now.
Okay, I have. Should I just read out what it says?
Sure, so depending on everybody's, I mean, I'm happy to do it if people have time.
No, we can do it. I can do it a different time.
You can put it in the chat. Why don't you put it in the chat, Tina, and then I can respond to you. Good plan.
Perfect. So at this time, we'll transition and have Dr. Lemke take over and present on GRI disorders and registries and all of those fun things.
Yeah, so let me try to share my screen. So can you see the presentation in presentation mode?
Yes.
Perfect. Yeah, so thank you very much. My name is Johannes Lemke. I'm a clinical geneticist from Leipzig in Germany. And I have been involved for about 10 years now in GRIN disorders and genetic research on GRIN disorders, and also in diagnostics. And I would like to give a presentation on GRI disorders, not only GRIN, but GRI. I would, as an introduction, I would briefly just introduce our team. I would like to just briefly explain what GRI actually stands for. What does it mean? I would say a few words on how common GRI disorders are. I would like to, that's a main part of the talk, give you some impression on what is the clinical presentation of GRI disorders. In particular, are there similarities or differences among GRI-related disorders? Are there any symptoms that can be predicted from a particular variant that had been detected in the genetics report? And is there anything that we can say on the long-term outcome on those patients? And finally, what is the sense and what is the help of a registry of GRI patients? So yeah, let me just briefly start. I have a, how can I, that way, okay. That's the team of ours, my team in Leipzig. Leipzig is positioned right in the middle of Germany, more or less. And we have a few colleagues a few of them already have worked for a few years on GRIN disorders, basically. There's Eriskay, who has a bit of a focus right now on treatment of GRIN disorders, and also is involved in the registry that we are maintaining. We have Chiara Klöckner, who is a medical student, who also is involved in the registry, but mostly she was the one that contributed very much to the GRIN portal that you may have seen already. And we have Konrad Platzer, who is a resident. No, he's not a resident any longer, sorry, he's a consultant. Also in clinical genetics. And he has worked a bit particularly on GRIN2B. And Vincent Strelo, who is also a consultant, who has worked on GRIN2A. And so that's our team. And yeah, so let's start. What are GRI disorders, and what does GRI actually mean? So GRI is an acronym, it's an abbreviation for glutamate receptor ionotropic. Ionotropic means that the glutamate receptor is actually an ion channel which is ligand gated. This means that there is a ligand, glutamate in this case, that binds to the extracellular part of the protein of the receptor, and this binding of the ligand leads to an opening of a channel pore and allows ions to pass through this ion channel from the extracellular space to the intracellular compartment. And there are 4 different types of these ionotropic glutamate receptors. One is the N-methyl deaspartate type, difficult word, and the short form is NMDA, and it starts with an N. That's why it's this part of glutamate receptors are called GRIN, NMDA. The second subtype is specific to AMPA, which is an even harder to pronounce word. I want to try it. It starts with an A. And then there is the kyanite receptor leading to the GRIC genes and the GRIC subtype of the glutamate receptor, and the delta, which is the GRID. I think I didn't say that the AMPA is then actually the GRIA subtype of the receptor. So that's why we have GRIA and GRIN and GRIC and GRID, and they all together are actually the GRI subtypes of the glutamate receptor. Glutamate itself is the most prominent neurotransmitter in the brain. So it is necessary for the cells in the brain, for the neurons to communicate with each other. And normal glutamate signaling is very important for normal brain functioning and for normal brain development. So if there is an individual that has a pathogenic variant in any of the GRI genes, then this may potentially lead to a dysfunction of the particular receptor, and a dysfunctional receptor will lead to a dysfunctional glutamate signaling, and this will lead to an abnormal brain development, which will have consequences such as developmental delay, intellectual disability, epilepsy, or movement disorders, ataxia, psychiatric disorders. There are various possibilities of disorders that can arise through these dysfunctional glutamate receptors. So one question that I have been asked quite a few times is, how often are GRI disorders, or how frequent are they? There is not a simple answer to this, or actually there is one simple answer, and the simple answer is that it is very rare, but that is not a very satisfactory answer. There are 2 ways how to approach this question. One is a hypothetical estimate. There is an algorithm that has been developed by the group of Dennis Lull from Cleveland, where you can predict the probability of a gene to have a de novo variant. And if you apply this algorithm for a few disorders, which I did here in this table, and in the 3rd line there are the GRIN genes, then you can see that, for example, for GRIN2B, that's here, you would expect, hypothetically, that there are about 220 newborns in the US, according to the newborn numbers in 2018. So 220 newborns with GRIN2B, with GRIN2B de novo variants. That sounds like really a lot. But you have to keep in mind some biases. First, not all of the de novo variants are pathogenic. So you can have a de novo variant in GRIN2B, but not have a phenotype at all, because the variant perhaps is not pathogenic at all. It's just a rare variant. That's not very often, but this can happen. It can very well happen that the de novo variant in an individual is not being diagnosed because the patient has not undergone genetic testing for whatever reason. There are multiple possibilities why a patient has not been tested genetically. There's also a risk of premature death or even prenatal death. That means perhaps, also this is a hypothesis, perhaps the disorder is so severe. For example, it could lead to a malformation of brain development and a cortical malformation that the fetus is not able to survive until birth. So the fetus will not be a live birth, and the pregnancy may get lost on the way. And if there is a pregnancy loss, we perhaps will never know what the reason for this was. And of the thousands and thousands of pregnancy losses alone in the US per year, we don't know how many of them may perhaps be due to GRIN2B or any other GRIN disorders or any other GRI disorders. We can only speculate, but we certainly do not know. Another reason for lower number that we feel are around is maybe there are also several families that will not connect, that will have a genetic diagnosis, but will not reach out to Cure GRIN or to other patient organizations and will not connect through Facebook because they do not want to talk about this diagnosis with other parents. Could very well be. We have seen this quite a few times. And most importantly, all of this is just a hypothetical estimation. So this is not a calculation that is truly valid. It's nothing more than a sophisticated guess. And currently we do not really have something to really validly predict the numbers. What we have is we have the numbers of individuals that have registered in the registry. So we can tell you how many patients we know of. And what we know of is there are around 200 individuals with GRIN1 variants, nearly 200, more than 250 individuals with GRIN2A, about 200 individuals with GRIN2B, and less than 20 individuals with GRIN2D. That's only for the GRIN genes. For the GRIA and the GRID and the GRIC, we are even less than GRIN2D. And so we do not know a lot. We know a little bit on GRIN1 and 2A and 2B. And only because we know that there are many GRIN1, 2A and 2B patients, and of several of them we do have phenotypic information, we can extrapolate some clinical data. And this is something very important for genetic counseling and for newly diagnosed patients. So let me give you a brief overview just on GRIN2A on this slide, just for an impression. So I choose GRIN2A because GRIN2A is the gene where we had the most individuals and the most comprehensive phenotypic data. And that was a gene we published on 2 years ago, or actually already 3 years ago now. Yeah, it had been worked up most comprehensively in comparison to all the other GRIN genes currently. So what we know is that, as you can see in the chart A, this pie chart here, intellectual disability and developmental delay. We had this information on 177 people. And we know that more than a third of these people had no intellectual disability and no developmental delay. So they had a normal intellect, which was surprising. That is actually a significant number. There are a few individuals that had mild intellectual disability, even less with moderate, even less with severe, and a few with profound intellectual disability, and also several cases where we know there was intellectual disability, but we didn't know the degree of how severely affected they were. However, just roundabout 1/3 was not affected by intellectual disability, 2/3 had intellectual disability. Epilepsy, just a minority of just over 10% had no seizures and no epilepsy. All the others had epilepsy. So epilepsy was a really frequent feature seen in more than 200 individuals. Or so 200 individuals were the overall, and about 90% of them had epilepsy. This means you could have a GRIN2A disorder and not have intellectual disability, but very well have epilepsy. And also, if we go to pie chart D, we see that only 8% of individuals have normal speech. So it is very likely that you can have normal intellect, but still have a speech disorder, perhaps in addition to epilepsy. EEG was also abnormal in most of the patients. Only 6% of individuals had no EEG abnormalities. That means also, again, you could have EEG abnormalities, but still do not have seizures. So that's for GRIN2A. And yes, for GRIN2B, I didn't have these pie charts, but for GRIN2B, there is one image that is particularly to be pointed out, which is the brain MRI. You can see here on the left side a normal brain, and a little minority of GRIN2B patients and also of GRIN1 patients will have a particular pattern of a brain malformation. So for every one of you who's not very familiar with brain MRI, you can basically say that a human brain should more or less look like a walnut. And so if you think this is a walnut and then you look at this, you see that the surface of this walnut doesn't look right. That's why are these white arrows. It's the same here and the same here. You can also see that the inner spaces of the brain, not of the walnut, they are enlarged in most of these cases. So much broader and bigger than in the normal situation. A third abnormality is something called the basal ganglia. That's a little structure which is in the middle of the brain, which is located here, should be of more or less of this size. And you can see all the patients have enlarged basal ganglia. The structure is bigger and more prominent, marked with an asterisk in all of these cases. And so in summary, you have this abnormal surface called polymicrogyria, you have enlarged inner spaces, and you have dysplastic and enlarged basal ganglia. So that's a really complex and severe brain malformation, which also appears to be quite typical for GRIN2B and GRIN1. So if we compare all the GRIN genes with each other, and I'm currently only talking about GRIN, then we know about intellectual disability that the vast majority of individuals with GRIN1, as well as GRIN2B, as well as GRIN2D, they are severely, moderately, severely, or profoundly intellectually disabled. Mild intellectual disability in GRIN1 or GRIN2B, and likely also GRIN2D, is something very, very rare. A little more prevalent in GRIN2B, but very rare in GRIN1 and almost never seen in GRIN2D. Although the numbers of GRIN2D individuals with clinical data is very low, as I have shown you in the prior slide. What is exceptional, I already told you, that more than a third of individuals with GRIN2A related disorders have normal intellect and no intellectual disability. That's an exception which accounts for all the GRIN genes. As far as I'm aware, there's no other GRIN genes, no other GRIN disorder other than GRIN2A where affected individuals can have normal intellect. If we look at seizures, that is a very prominent feature in all of the GRIN disorders. So 70% of GRIN1 individuals will have seizures, about 90% of GRIN2A individuals, half of the GRIN2B individuals, and nearly all of the GRIN2D individuals. And there's a particular epilepsy syndrome, which is called epileptic spasms, which is a part of so-called West syndrome, which is a which is an epilepsy type, which has been seen in a small fraction of GRIN1 cases, exceedingly rare in GRIN2A, rather often in GRIN2B, and very often in GRIN2D. And GRIN2A is also known for other epilepsy phenotypes, which are distinct, but not as severe as, for example, the epileptic spasm type of epilepsy. If we look at speech and speech impairment, then we see that it is most prominent in all of the disorders. Only in GRIN2A, there are a few people that will not have a speech impairment, but actually this is a bit a biased calculation, because of course, if an individual is severely intellectually disabled, this individual will have a speech delay and will never achieve normal language as you and I have. So of course, they have a speech developmental delay. Particular for GRIN2A is that they also have a speech disorder. They have a distinct pronunciation, a distinct verbalization. They have difficulties to pronounce vowels. So there's a real a speech disorder other than speech developmental delay. Movement disorders is also something that is seen quite frequently in GRIN disorders, very often in GRIN1 and also in GRIN2D, rarely in GRIN2A and sometimes in GRIN2B. For GRIN1, it is to be mentioned that there is a particular type of a very peculiar movement disorders called oculogyric crisis, which is a movement disorders of the eyes when they, for example, move upwards and do not move as they should. That is a feature that is not actually really seen in many other disorders and is thus rather specific, actually, even for GRIN1. If we look at muscular hypotonia, so the muscle tone and the a bit also the strength of these patients, then we see that hypotonia is quite prevalent in GRIN1 and 2B and in 2D, and something very rare in GRIN2A, which we also would expect because there are so many individuals that do not have developmental delay and that do not have intellectual disability and that actually develop quite normal, with the exception of seizures and perhaps speech development. If we look at the MRI, then we know that several individuals with GRIN1 can have an unspecific abnormality called cortical atrophy. That means that the spaces of the brain fluid are a little bit enlarged and the brain looks like it is, has been shrinked a little bit. And in some individuals, a very small minority, we can see this peculiar brain malformation, which I have shown you earlier, which is the identical brain malformation as seen in GRIN2B, where it is a little bit more prevalent, with 14% of individuals having this malformation. On the other hand, fewer individuals with GRIN2B have this cortical atrophy. Cortical atrophy is also rarely seen in GRIN2A, and it's always seen in GRIN2D, but again, numbers are very, very small of the individuals where we know the data of the brain MRI. Is there anything that we can predict from the variants? Yes, there is. If we look at the gene down here, then you can see the black bars are the variants that are known to be found in the healthy population. So every one of us could have any variant which is marked down here. These bars up here, these are the de novo pathogenic variants that have been observed in affected individuals with GRIN2A related disorder. And you can see on the one hand, for example, this last section of the gene, CTD, which is the C-terminal domain, an intracellular part of the glutamate receptor, this part of the gene carries really a lot of genetic variation, which is quite prevalent in the healthy population. So this is apparently quite tolerable to genetic variation. This all refers to missense variants, by the way, not to nonsense or truncating variants. So we see a lot of missense variants in the healthy population, but we do not see one single pathogenic missense variant in this part of the gene. So possibly there are no pathogenic missense variants back there in the gene. It looks like all the pathogenic missense variants cluster in this middle part of the gene, in the ligand binding domains, LBD, in the ligand binding domain S1 and S2, as well as the transmembrane domains, particularly M2, M3, and also M4, and a little bit in the linker sequences in between. And there are very rare exceptions of variants which are located in the extracellular part, in the amino terminal domain. There are just these 3 exceptions here. So if any individual has a missense variant that aligns to this model, then we know if individuals that have a missense variant in the amino terminal domain, there are only 8 individuals. Some of them have recurrent variants, so there are 3 different variants that we know of. They all have either no intellectual disability, the degree of severity of intellectual disability is 0, or they have mild intellectual disability. So they are rather mildly affected. That's a bit similar to the individuals with missense variants in the ligand binding domain S1 and S2, which you can see here and here. You see that most of these individuals have either no intellectual disability or are mildly affected, and very few exceptions have moderate or severe intellectual disability. And by contrast, you see here the M2 transmembrane domain and the M3 transmembrane domain, where here all individuals with pathogenic missense variants in this domain have profound intellectual disability. Here as well, all have profound, with one individual that has only severe intellectual disability. And also the linker regions between the transmembrane domains are usually affected by profound intellectual disability. So what we can say already from the variant, if there is perhaps a newborn that has been diagnosed and we do not know about the developmental perspective, we can already say, okay, the variant is located here or there, so therefore we expect that there is a high probability of a rather mild phenotype, or we know the variant is located in the M3 domain, and we expect this looks like it will perhaps be a very severe phenotype. So we already know a few things that we can predict. If we compare GRIN2A to GRIN2B, then we actually see a very similar pattern. This one is GRIN2A, which we have just seen in the prior slide. This one is GRIN2B, so we see the same clustering of variants in the healthy population, and we see the same clustering of pathogenic missense variants in affected individuals. And there are 2 unclear variants back here, which we are not sure about whether they are pathogenic or not. Perhaps they are not pathogenic. So I've only been talking on GRIN so far, but I have a few words to say on the other GRI disorders, particularly on GRIA. The phenotype of GRIA disorders also comprises global developmental delay, intellectual disability, poor or even absent speech, ataxia, dystonia, so movement disorders, behavioral abnormalities, for example, autism spectrum disorders, sometimes regression. Half of the patients develop epilepsy, which is very often treatment refractory and very difficult to treat. And a few patients have died already in infancy. And so this is something that also we have been asked quite a few times, is there regression in GRIN disorders, which has been described in GRIA? And what is the life expectancy of GRIN patients? So both are really difficult questions. First, for the regression, As you have seen earlier, we are aware of quite a few hundred individuals with GRIN-related disorders. And we know of maybe a small handful of individuals that apparently had regression or signs of regression. However, it looks like this is not a dominant feature and it's something exceedingly rare, but perhaps it could happen in some severe cases. We do not yet know which are these cases and which are predictors or which are features that can help us predict that this patient is susceptible to regression. And also to the life expectancy, we do not have a lot of data on older individuals. Most of our individuals, I will show you a graph on this in a few minutes or a few seconds. Most of the data we have is on children and, let's say, teenagers. We barely have any data on adult patients, which is a problem that we have for actually all developmental disorders and all genetic conditions with developmental delay. If you look at GRIA3, Keith already had posted in the chat that this is the only gene which is located on the X chromosome and is thus an X-linked disorder and will predominantly affect males, in contrast to all the other GRI genes where both males and females are affected similarly. Also in this condition, there is global developmental delay, intellectual disability, speech impairment, behavioral abnormalities, and epilepsy. So again, it's more or less the same spectrum of disorders. GRIC2 is another example. Again, global developmental delay and intellectual disability, speech impairment, ataxia, behavioral abnormalities, and epilepsy. Again, just the same spectrum as the others. We do not really know, let's go back. We do not really know the precise spectrum as I had shown you for GRIN2A in one of the first slides, because we do not have 250 patients where we can really destruct the proportions and show you, like, for example, 88% have epilepsy and 12% do not have epilepsy. We cannot say for GRIC2 because we barely have, we have less than 10 patients. So we cannot do these calculations yet. What do we know about the long-term outcome? I already had told you just before that most of the data we have is on children and teenagers. So this is from a genotype-phenotype correlation that we currently perform on GRIN1. So we would like to repeat what we had done for GRIN2A a few years ago. We would like to repeat for GRIN1 right now. And we have a cohort of about 180 individuals. Of a little more than 100 of them, we do have comprehensive clinical data. And as you can see here, that's just the age distribution. Most of these individuals are toddler, early childhood, middle childhood, adolescents, and only 5% of individuals are older than 18 years. So we barely have any adults. Thus, we cannot really say much about the long-term outcome. We do not know. Still, we really would like to know, and we need to know to be able to tell you what is the age of those individuals that have died. So we know that there were a few individuals in the past few years that even had been connected with Cure GRIN, which had unfortunately already died. And even these individuals are most important for our statistics because we would like to know what age had they been, what reasons had they died of. Was there a recognizable reason, or was it a so-called sudden unexpected death in epilepsy, so-called SUDEP? Or was there anything else that would be relevant for this individual? So the more individuals we know of, the more data we have, the better we can correlate these data, and the better we are able to counsel newly diagnosed individuals in genetic counseling and to provide you with the data. And yeah, and that's actually the reason why I think the GRIN portal, and in particular the last part of the GRIN portal, the GRIN registry, is most important. This has been designed as a GRIN portal and a GRIN registry, but we already opened the registry to a GRI registry. And we will also broaden the scope of the GRIN portal to a GRI portal in the near future. But what I would like to point out is the more individuals, the more patients register in this registry, which is a global registry, which is hosted by several research groups around the world. The more individuals register, the more data we will have, and the better we will be able to counsel you and to really understand and to delineate the phenotype and to help finding cures actually for GRIN disorders and GRI disorders. And I think with this, I'm at the end of my talk and I'm happy to take questions. Thank you very much.
Thank you, Dr. Lemke. Are there any questions from the audience?
I just realized it got dark and I can't be seen any longer.
I don't see any questions in the chat. Feel free to unmute yourself if you have any questions for either Eriskay or Dr. Lemke.
So just to clarify, Dr. Lemke, the registry now, anyone with GRIN or GRIA can participate, and you hope to open it up soon to GRIC as well? Is that right?
The registry is already open. It is still called GRIN registry, but if you participate and you log in, then you can also choose all the other GRIA, GRIC, GRIT genes. So it's open to anyone. We already have a number of GRIA individuals. We collaborate with this with a group from Alan Bayat from Denmark, who's actually hosting this group, let's say. So he's the main investigator collecting this data. And we will, or our aim is to display the results of all the GRIA patients, and if possible also of GRIC and GRIT, on the portal as well, which we had done for GRIN already. So the first step is to broaden the registry from GRIN to GRI, which we already had done. We just have not named it yet like that. And the second step will be to broaden the GRIN portal to a GRI portal and display all the GRI data.
That's great. And it's the same, it's open currently, whether it's the North American version through Dr. Benke or your version.
I think yes and no. For GRIN, it stays the same. For all the others, we haven't really discussed, and I think because GRIA is actually mostly collected by the Danish group and has not been so far really collected a lot by Tim Benke. I think it would be the easiest way to enter it in the online registry. But we will put it the correct way on the website as soon as we have discussed with everyone. But in the end, we all collaborate with each other, and yeah, so we will share the data anyway.
Perfect, thank you. See, Lana has her hand up.
Hi, I have a question about the ages identified. That one slide, I was wondering if that was really a function of when the genetic testing became more widely available. You know, that in the older kids, there's not that many identified. And is that because there wasn't that testing available?
Absolutely. That's the exact point. So if a child is diagnosed with developmental delay, with epilepsy, or a developmental problem, which manifests in childhood, then that's the time where also people think of maybe we should do genetic testing. And so if this happens now, or if this had happened 5 years ago, this child may have gotten comprehensive genetic testing. If this was 10 or 20 or 30 years ago, there was no possibility of comprehensive genetic testing, so this had not been performed. And this child is possibly now 30 or 40 or 50 years old, is possibly considerably intellectually disabled, and possibly lives in a residency of individuals with intellectual disability, and has the life is set there, and there are people taking care of this individual, but they're likely not people that are really asking what is actually the diagnosis of this individual. So these people live in their residencies and they have their life, but we do not know the diagnosis. And as long as we do not know the diagnosis of these elderly individuals, we will not know the course of the disorder.
Right, then it makes it challenging for parents when you see life expectancy. You know, all we know is that it's unknown,
yes.
Right?
We know that it is unknown. We know that it is very likely decreased, as for actually all of developmental disorders with a considerable delay. And we know that it is likely decreased because we already know of a few individuals that had died suddenly in childhood or in infancy or during the teenage age. So we know of individuals that have died, which is, which points towards a decreased life expectancy, but we do not know, do they get 30 or 40 or 50 or 60 years old or even older than this? We do not know.
Okay, thank you.
You're welcome.
I just had a question. Is it typical for kids with GRIN to have the variant, the KCNQ as well? Or is that just an example you were using in that one slide?
No, that was just another example. So normally you either have a GRIN disorder or you have a KCNQ disorder or you have a CHD disorder. That were just a few gene families that are used as examples. So they are not related to each other.
Okay, because our son has both. That's why I was just curious.
Sorry, I didn't understand that.
Our son has both, the GRIN1 and then the KCNQ3. So that's why I was just curious about it.
Oh, really?
Yeah.
Okay, and both are confirmed pathogenic?
No, one was pathogenic and the other was of
variant of uncertain significance?
Yes, yep.
Yeah, then the pathogenic is likely the cause of the disorder, and the variant of uncertain significance is likely, let's say, just a variant that couldn't really be classified, but it's perhaps not really the cause or also not a modifier. It was just an unclear variant at that time of the report. It should be re-evaluated in 1 or 2 years, maybe.
Okay.
I just wanted to add, I think maybe to both of those points, that I think we're, you know, at a place where we're trying to, in the genetics field anyway, trying to increase access to, you know, technology like exome and genome sequencing to patients earlier in the journey of the diagnosis and of even just the symptoms, you know, because we, you know, to the first person who asked a question there, I think that's you know, an issue is right now some of the technology is just not available to people, and so they, you know, present with, you know, symptoms and then go sometimes years without really understanding or knowing why, which can be difficult and challenging for families and the patients, but also just from a management and treatment, you know, especially as we learn more and more treatments are available, this is something that you want to do, you know, as early as possible. So, you know, hopefully in the not too distant future, we can start, you know, using these technologies to support a more efficient and earlier diagnosis for patients, not just with, you know, in the GRI category, but for all patients with underlying genetic diseases.
And I also just wanted to add, because I think for me, some of what Dr. Lemke said feels scary just in terms of the life expectancy stuff. And I think it's, I think Dr. Lemke, you mentioned, we don't know if it's 30s, 40s, or 50s, but I think it's like when we think about life expectancy at birth, generally we know that that's lower for people with intellectual disabilities. But it could be that for people who live, to 20, that the risks become less. Like it's not impossible that a life expectancy could be really dragged down by people who die early on, but that people who live till 20 could well live into their 60s or 70s. We see that in some diseases. So I think for parents, there's two things. One, I don't think we should get overly worried about life expectancy, but the other is we still need to plan for our kids after we're not around. I don't think we can assume that they're not going to outlive us. Exactly, that's an important point. I am for GRIN1, at least I'm aware of one individual from Switzerland who is in his 50s or even around 60 years old. So they can get old. And he lives in a residency in Switzerland of people with intellectual disability. And of course, that's an age where the parents cannot take care of this individual any longer. So they will be in their 80s and maybe are not even around any longer. So yes, it may certainly happen that these individuals get so old that they will survive the parents. So it needs to be planned on how they will be taken care of at a point when the parents cannot do so any longer. Okay, I think, thank you very much for all these questions.
All right, so I don't see any questions in the chat. Any last minute thoughts from anybody? Okay, so I think this concludes our first lesson. So again, we're going to reconvene on March 26th as well as April 9th. On March 26th, we're going to talk about neuroscience, the receptors themselves, and learn a little bit more about those and keep building on things and talk about personalized research. And then in the last session, we'll talk about GRI treatments and cures. And I just wanted to take a second to thank Dr. Lemke and thank Eriskay Liston for your time. And thank you all for all of your attention today. We greatly appreciate all of your questions, and we'll see you in a couple weeks.
Just before we go, I just also want to thank Megan. She did an amazing job putting together all of these sessions for us. And I don't know, today was great. So thanks to all three of you, really appreciate it, and looking forward to the next two.
Thanks,
have a good rest of the weekend, everyone.
Thanks, everyone.
Bye, thanks.
Take care.